You added a leaf, a catappa, maybe an oak, or maybe even just a piece of driftwood. Something organic, something that was alive once, something you placed in this tank on purpose. And now it's disappearing. Not all at once, not in a way that you can watch in real time, but incrementally, consistently, in the direction of gone. And you probably never thought very hard about it exactly what is doing this. The leaf did not disappear on its own. This is feeding. This is the opening act of the entire decomposer food web. And we somehow did not introduce it until now. This is the Shredder Guild, and the episode is theirs. The amphipod is a crustacean, same class as shrimp, same class as crabs, the same broader phylum as every jointed-legged animal we have covered on this channel. The family resemblance is there if you look for it. The execution is unusual. It swims on its side, not as a choice. Its body is laterally compressed, flattened sideways rather than dorsoventrally, which makes upright swimming geometrically awkward and horizontal swimming perfectly natural. The amphipod has committed to this orientation completely. When it is not swimming, it is feeding. 7 pairs of walking legs grip onto the surface while the mouth parts, the gnathopods, modified appendages at the front of the body, rasp and tear at leaf tissue. This is systematic, sustained, purposeful shredding. The female carries her eggs and young in a marsupium, a brood pouch formed by modified plates on her underside. The young emerge as miniature adults, which is, as this channel has noted with increasing frequency, simply what crustaceans do when they are being efficient about it. In a healthy established tank, amphipods are everywhere. They are under the leaf litter, in the mulm, along the driftwood, in the crevices of hardscape you've never looked at closely in months. They are almost certainly in your tank right now, and you have almost certainly not noticed them. And they're almost certainly fine with that. The isopod is also a crustacean, also technically related to the shrimp in the tank, though the relationship is distant enough that both parties would probably prefer not to dwell on it. Where the amphipod was laterally compressed, flattened sideways, the isopod is dorsoventrally compressed. It is flattened top to bottom. It is essentially a small armored oval that moves across surfaces with confident hurriedness of an animal that has been doing this since the Carboniferous period. The segmented plates along its back are called tergites. Each one overlaps the next in a design that provides flexible protection without sacrificing any mobility. The same engineering principle in every woodlouse, pillbug, and roly-poly you've ever encountered, refined over approximately 300 million years, and apparently deemed satisfactory by nature. It feeds in the same manner as the amphipod, rasping at leaf tissue with modified mouthparts, working across the surface systematically, with a composure that suggests it has done this before and intends to continue doing it long after any particular observer loses interest. Which is accurate. Isopods were shredding leaves before there were fish to ignore them. Freshwater isopods in the aquarium are generally found lower in the substrate and in the mulm layer than amphipods. Deeper, darker, more committed to the sediment. They appear, they do their work, and disappear back into the material they came from. The tank is better for having them. They are indifferent to this fact. In stream ecology, organisms that feed on leaf litter and woody debris are assigned to a functional feeding group called shredders. What they do to the material, not what they are. The leaf that enters this tank is coarse particulate organic matter, CPOM. It is large. It is tough. It's cellulose and lignin, and it is chemically resistant. Most of the organisms in this food web cannot eat it directly. It is too big, too complex, and the shredder's job is to change that. As the shredder feeds, it fragments the leaf, mechanically, physically, tearing the CPOM into pieces small enough to drift into the water column, into the substrate, into the reach of the next tier of the food web. Fine particulate organic matter, FPOM. What was the leaf is becoming food for organisms that could not have eaten that leaf. The Neocaridina shrimp in this tank are collectors. The Cereodaphnia in the culture jar are filterers. They are waiting for the shredder to do the first step. Without the shredder, the leaf sits. It ages. Bacteria begin to soften it, slowly. But the fast root, the efficient root, the root that puts the energy in the leaf into the food web within days rather than months, that root requires a shredder. And the shredder does not retain all of what it processes. Much of what passes through its gut is excreted as fecal pellets. Small, dense, microbe-rich packages of partially digested organic matter that settle into the substrate and become food for a different set of organisms entirely. The shredder feeds the food web twice, once through fragmentation, once through defecation. It is not glamorous, but assuredly essential. In our Detritus episode, we built a map of the decomposer food web. 4 stages: leaching, conditioning, shredding, microbial mineralization. We named those stages. We described the pipeline. We were, in retrospect, describing the work of the animals in this episode without introducing them. Stage 1. Leaching. Water-soluble compounds leave the leaf within the first few days. Tannins, sugars, simple organics. The water turns colors. This is the easy part. It's chemistry, not biology. Stage 2. Conditioning. Bacteria and fungi colonize the leaf surface, beginning to break down the cellulose and lignin matrix. This makes the leaf softer, more permeable, more nutritious, and more attractive to the animals that perform Stage 3. Stage 3. Shredding. The conditioned leaf is now physically fragmented by the shredder guild. Amphipods Isopods. The animals that take what bacteria prepared and make it available to everything downstream. Stage 3 is this episode. Stage 3 is what we have been building towards since Detritus placed a catappa leaf in this tank. Stage 4. Microbial mineralization. The fragments become FPOM. The FPOM is colonized and broken down further. The nutrients return to the water column. The cycle completes. Your leaf is gone. The tank is richer for it. And somewhere in the substrate, the shredders are already working on the next one. We do not know what genus of amphipod this is. We do not know what genus of isopod. The identification of freshwater amphipods and isopods to the genus level requires microscopic examination of mouthparts and appendages under conditions that are difficult to replicate in an aquarium setting. And we didn't do that. The amphipod, for its part, doesn't know either. It's just feeding. It finds this sufficient. But here's the thing about functional ecology. It does not require a species list. The EPA's benthic macroinvertebrate bioassessment protocol, the method used to evaluate stream health across the United States, works primarily with functional feeding groups, not genera. The shredder population in your tank is not something you measure with a probe. Lift a piece of leaf litter. Look at what moves when the light hits the substrate. A dense, active shredder population is a sign that this tank has what they need. Leaf litter as a food source, sufficient dissolved oxygen in the lower water column, and enough structural complexity in the substrate for them to shelter and reproduce. It is a bioindicator of a functioning decomposer food web. The shredders are present because the conditions that support them are present. A sparse shredder population in a tank with leaf litter suggests one of three things. Dissolved oxygen in the lower water column may be insufficient. Shredders, like most aerobic organisms, need oxygen, and the substrate's deepest layers are the first to go hypoxic in an overstocked or under-aerated tank. Or the leaf litter input may be insufficient, not enough food to sustain a population, or possibly both things. The third possibility is one that aquarists discover last. The tank may be too clean. If the gravel vacuum runs every week, if the leaf litter is removed before it can condition, if organic matter is systematically eliminated as fast as it accumulates, the shredder population has nothing to work with. The decomposer food web does not operate on a substrate of pristine gravel. It operates on material. Give it material. Every decomposer food web has a beginning, a first step, a mechanism that takes the material that falls into the system, the leaf, the bark, the dead plant matter, and makes it accessible to everything that comes after. Leaves do not simply stay as large, intact pieces in the tank. The shredders do the first visible bits of renovation for what will become the most diverse habitat in the tank. It is these animals, Amphipods and Isopods, and the rest of the shredder guild, working in the substrate in the dark, doing the first things that everything else can do the second thing. They mostly go unnoticed by the inhabitants of the tank. They are not the most studied by the observer of the tank. They are likely not the creatures you bought or placed there deliberately. They arrived on the leaf litter, on a plant, or in the mulm, or perhaps even filter media from another tank. And they stayed because the conditions were right, and they have been working ever since. You added a leaf. You didn't know who would find it. You didn't need to know. The shredders found it first, as they always do, and they have been doing in freshwater systems long before there were aquarists to not notice them.